Hello, thank you for attending my talk today. My name is Justin Conover, and I'm currently a postdoc at Iowa State University. And today I'm going to show you some of my work from my dissertation, where I found that deleterious mutations accumulate faster in Allopolyploid than in Diploid cottons. One of the most ubiquitous findings in plant biology throughout the genome era is the importance of whole genome duplication or polyploidy events throughout angiosperm evolution. If you were to take a phylogeny of all the angiosperms, you would see whole genome duplication events pop marked throughout the entire phylogeny, as shown here, including one event that predates all angiosperms. Polyploidy is so widespread in the angiosperms, in fact, that only last year, the genome sequence of Aristolochia was sequenced. And it was the second species that showed zero evidence of any whole genome duplication events since that event that predated all angiosperms. Although polyploidy is important in angiosperm evolution, there are still a lot of important questions that we don't have answers to, such as how does evolution act differently in polyploids compared to diploids? And today I want to focus on one aspect of that, and that is deleterious mutations. Now, a lot of work has gone into population genetics to determine the factors that influence how deleterious mutations accumulate or are purged. Some of those include the effect of population size, recombination, life history strategies, and of course, genetic dominance. However, I think that there's a missing aspect in plant evolution that may be driving how deleterious mutations accumulate, and that is gene and genome duplication. In 1932, J.B.S. Haldane was the first scientist to make this hypothesis. And this question, even though it's 90 years old, still has not been robustly tested. But today I want to show you some of my work in the cotton genus in Gossypium that may shed some light on this question. So most of us know cotton as the species that makes all of our clothing and fibers, but actually that species is only one of a handful of allopolyploids from the genus Gossypium. And those allopolyploids formed from the hybridization of an ancestor of Gossypium remundii, which is located in northern Peru, denoted by the species abbreviation D5, and an ancestor of the A1 species, Gossypium herbaceum. And this hybridization happened about 1 to 2 million years ago, even though these two diploids diverged between 5 and 10 million years ago. Since that initial hybridization event, they've since diversified into what's currently 7 recognized species, including Gossypium mustelinum, or 84, that's found in Northeast Brazil, 82 and 85, that's found in the Galapagos Islands and in northern South America and Central America. And then there's another clade that includes a cotton species in Hawaii, and as well as Gossypium hirsutum, which is the species that we all think of as traditionally cotton that makes all of our clothing, and 2 very closely related species, 86 and 87. So one way that we can use comparative genomics to understand deleterious mutations is to look at a multi-species whole genome alignment to figure out those positions in the genome that have been phylogenetically conserved over deep evolutionary times. Now, the rationale behind this is that if there is a mutation in your data set that occurs at a phylogenetically conserved position, that's most likely to be deleterious because mutations are a random process, and somewhere throughout this phylogeny, there likely was one or multiple mutations at this position that has been subsequently removed by purifying selection. One of the first steps in comparative genomic analyses is to curate a list of single copy genes between all of your species of interest. However, because of the prevalence and importance of polyploidy in plant genomes, a lot of the most commonly tools for this task don't explicitly incorporate ploidy into their estimates for inferring orthology. So I developed a tool called Phytogenic, which uses synteny and explicitly knows the difference between different ploidy levels to extract these sets of single copy genes, shown here in blue. However, because of the ancient polyploidy events throughout the angiosperm phylogeny, you will also not only see the single copy orthologs, but you can pull out these ancient collinear blocks that are the result of ancient polyploidy events as well. Phytogenic is a Python program, and all of the code is publicly available on GitHub. And if you scan the QR code at the bottom right hand of your screen, that will take you to the GitHub repository to check it out for yourself. And this can be used for any ploidy level, including only diploids. If your analysis doesn't include any difference in ploidy between your species, but you want some sort of syntenic relationship to have higher confidence in the orthologs that you are inferring. So this takes us to the deleterious mutations in Gossypium. At the top right of your screen, you'll see the map showing the two diploid species in red and black, and the other colors represent all the polyploids, and those colors will be similar in the graphs that I show you as well. For this analysis, I used almost 9,000 pairs of homologs that were determined by Phytogenic, and only used those homologs that had less than a 5% difference in their annotation length between the two subgenomes to try and minimize as much of the errors due to the differences in the genome assembly that we could. And when we look at the number of synonymous mutations that have arrived since the divergence of the diploid from each subgenome, we don't see much difference between the diploids and the polyploids. One thing to note here is that because the phylogenetic depth between the two subgenomes and their most closely related diploid is unequal, we will see a consistent difference between the two subgenomes here, but that shouldn't be taken as any biological interpretation, just that there is some difference in the phylogenetic depth to each diploid. When we expand our analysis to the non-synonymous mutations, we do see a difference where the polyploids have accumulated more non-synonymous mutations than either of the diploid counterparts. And then when we look specifically at those mutations that have occurred at phylogenetically conserved positions, here using GERP, which investigates sequence conservation at the nucleotide level, we again see an elevated number in the polyploids compared to the diploids. And when we also look at the protein level using a program called BAD mutations, we see a very similar result. If we were to restrict our analyses just to those mutations that have occurred since polyploid formation, we can also see a lot of the differences at the species level, where the purple species, which is the species located in Hawaii, tends to have consistently higher non-synonymous and deleterious mutations than the other polyploids, but it does not have an elevated rate of synonymous mutations, indicating that perhaps there is some founder effect for this migratory event to the Hawaiian Islands that could be leading to an increase in deleterious and non-synonymous mutations. If we were to look at the proportion of all the non-synonymous mutations that are deleterious, we also see a difference between the diploids and polyploids here. So this indicates that polyploids accumulate deleterious mutations faster than diploids do, due to that masking effect of having duplicated genes in the genome. And again, if we were to look at just those mutations that have arisen since polyploid formation, we see a lot of differences between species, and we also see a fairly consistent difference between subgenomes here. And again, this also indicates that these island endemics may have the highest load due to that founding effect. One of the more difficult tasks in doing this type of research is attributing this change in deleterious mutations to the effects of ploidy instead of to the effects of other factors that may influence the rate of deleterious mutation accumulation, particularly demography. Because polyploid formation is also accompanied by a really strong genetic bottleneck, that could artificially inflate the number of deleterious mutations, and this may not be a polyploidy effect at all. However, I don't believe that's the case in cotton, and I want to walk you through some of the expectations we would see if this was due explicitly to demography and what observations I see in my data. So if we were to look at a mutation that has a deleterious score of S, and we know what its effective population size is, anytime that deleteriousness is less than 1 over the effective population size, it's going to be controlled by genetic drift, according to the nearly neutral theory. And if that selection coefficient is greater than 1 over the effective population size, then it will be subjected to purifying selection and removed from the population. So if we see an instance where the affected population size decreases, therefore meaning more mutations are controlled by genetic drift, or if we see a population that is expanding and more of the mutations are controlled by purifying selection. And so for strongly deleterious mutations that have a high value of S, we shouldn't expect to see any change in the ploidy levels if this effect is only due to shifts in demography. However, for mildly deleterious mutations, depending on how deleterious they are in respect to the affected population size, we could see a lot of variability. However, when I can categorize these mutations into strongly deleterious, moderately, and mildly deleterious, we actually see that the strongly deleterious mutations are the most increased in the polyploids relative to the diploids. And we see this in both of the subgenomes and for every species that we're looking at. So this pattern does not fit the expectations we would expect under a demography scenario alone, and it suggests that the polyploidy is actually playing a role in this process. One potential consequence of this phenomenon is that DNDS estimations, when calculated between species of different ploidy levels, could be artificially inflated and may not actually infer weaker selection. So for this project, I work with Dr. Joel Sharbrough, who's an assistant professor at the New Mexico Institute of Mining and Technology. And we used 6 different allopolyploid systems, including the allopolyploid, its 2 diploid progenitors, and a phylogenetic outgroup. And we analyzed over 340,000 different genes from these systems. And we used orthology based on both synteny and on phylogeny. So we had a really curated, high confidence set of orthologs we can investigate. So for cotton here, if we look at the tree on the left with the 2 middle branches are the 2 tetraploid subgenomes with both of the diploid progenitors on either side. And if we look at the DNDS ratio, or omega as is shown here, we do see that there is consistently higher DNDS values for the polyploid subgenomes than for the diploids. And if we organize these species based on how old the polyploid is, we can start to see a bit of a trend where the older polyploids at the top tend to have significantly different DNDS values from their diploid relatives. And for those more recently evolved polyploids, such as Brachypodium on the bottom, there may not be that time yet for these mutations to have accumulated in a rate that's different from their diploids. Finally, I'd like to acknowledge the members of the Wendell Lab at Iowa State University, who I did this work with, including some of our collaborators at Colorado State University and Dr. Sharbrough at the New Mexico Institute of Mining and Technology, and of course my funding sources, including NSF and Cotton Incorporated. And with that, I would be happy to answer any questions that you have. Thank you for listening.